in biomolecules, the next type of biomolecule is carbohydrates. So carbohydrates are also the part of biomacromolecules which are having molecular weight more than 1000 Daltons, and these are also the part of acid insoluble fraction. Then these are actually the hydroxy aldoses and ketoses and their derivatives. So here in the aldoses it have the functional group called as aldehyde functional group. In the terminal part, that is CHO. Like we can take the glucose is one such example which contains aldehyde functional group, whereas ribose also. Then ketoses are the one which contains ketone functional group, that is in the middle or center, or we can say in the internal structure of the carbohydrate molecule. So in this one, C double bond O. So this type of structures can be seen in examples like fructose pseudoheptulose like examples. So likewise these are considered as the carbohydroxy aldoses and the aldoses, ketoses and their derivatives. So generally the carbohydrates have the molecular organization as CH2N or CN H2O-N. This is the molecular organization or the molecular formula, basic molecular formula by which the carbohydrates are represented, and these are generally considered as the saccharides. Saccharides are the one which are sugary in their nature. Then here we can see in the molecular formula, so here carbon, hydrogen and oxygen are the three elements which are present or by which the carbohydrates are made up of. But here always in this one, the hydrogen and oxygen are in 2:1 ratio. That is hydrogen number will be always double the number to that of oxygen. But of course some exceptionals are there, like in deoxyribose sugar, where the number of hydrogen and oxygen will not be showing 2:1 ratio. Whereas in all other saccharides, all other carbohydrates, we can see 2:1 ratio of the hydrogen and oxygen molecules. These carbohydrates are of different types based on the number of the monosaccharide groups which are present in them. So based on that one, we can say them as small carbohydrates. The small carbohydrates includes monosaccharides, monosaccharide derivatives, oligosaccharides are the three types, and the second group is the polysaccharides. The difference between oligo and polysaccharides is the oligosaccharides contains 2 to 9 monosaccharide groups. The polysaccharides contains at least 10 or more than 10 individual repeated units. So likewise that small difference can be observed between the oligosaccharides and the polysaccharide molecules. So in the small carbohydrates, let's start with the first one, the monosaccharides. Monosaccharides are the carbohydrates which cannot be further dissolved or further which cannot be divided to form into the further individual units. So this type of structures are called as a monosaccharides, and these monosaccharides are represented with C, N, H2N and ON molecular formula, or otherwise C, H2O, N, CH2O, N molecular formula. So likewise monosaccharides are the one which are not further can be divided into the smaller fragments. So many such examples are there, like glucose, fructose, such type of examples are there. So but here before that, before going to the glucose and fructose molecules, so based on the number of carbon molecules which are present in them, so the further classification are trioses, Trioses are the one which contains 3 carbons, that is basically represented with C3, H6 and O3. So trioses are the molecules which contains 3 carbons and 6 hydrogens and 3 oxygen molecules. In this one we can take many examples as glyceraldehyde and dihydroxyacetone are the examples of trioses. Tetroses. Tetroses are the one which contain 4 carbons in them, that is C4 represented as C4, H8 and O4 molecules, like in erythroses is the example of tetroses. Pentoses. Pentoses are the one which contains 5 carbons in them, that is C5, H10 and O5. So like ribose sugar, ribulose sugar, xylulose, such type of examples are there. But here deoxyribose is also there. Deoxyribose is the exceptional case where C4, H10 and O4 are present. So that is the only exceptional case where the formula, the molecular basic molecular formula is not exactly fitting to the deoxyribose sugar, because here the carbon number and hydrogen number is not in the proportionate to that of other monosaccharide molecules. Then hexoses. Hexoses are the one which contains 6 carbons in them, C6, H12, O6. So many such are there, like glucose, fructoses are many such examples. Glucose is the one which contains aldehyde functional group, whereas the fructose is the one which contains ketose functional group. So glucose is called as aldoses and fructose is the example for ketoses. Then heptoses. Heptoses are the one which contains 7 carbons in them, C7H14 and O7. So examples for them is pseudoheptulose. Pseudoheptulose are such examples where based on the number of carbons, so the monosaccharides are further categorized into trioses, tetroses, pentoses, hexoses and the heptoses molecules. Okay. The functional groups which are present, that is aldose or the ketose, they may form the bonds with or new bonds with either the alcohol or nitrogen. So when they form the bonds, those bonds are represented like this. When it is forming the bond with alcohol, CO, C bond will be formed, and if it is forming the bond with nitrogen, CN, C bond is formed. So both of them are called as a glycosidic bonds. The glycoside, that is the whatever the functional groups which are present, they will form the new bonds with some other molecules or some other functional groups which are present on the other organic molecule. Simply to say, the aldose and ketose of these monosaccharides will form the bonds with alcohol or the nitrogen of the other organic molecule. Such type of bonds are called as glycosidic bonds. The alcohol, the bond with the alcohol is represented with this, and the bond with the nitrogen is represented like this. So likewise they are able to form the new bonds. So here as they are able to form the new bonds, we consider them as a reducing sugars. So which can form the new bonds, the functional group which can form the new bonds, that is why we call them as a reducing sugars. All monosaccharides are reducing sugars. Pentoses and hexoses, they will exist both in the open chain form and also in the ring structures. In both forms they can exist. When they are forming in the ring structures, the two types of ring structures are there. One is called as pyranoses and other is called as furanoses. Pyranose structure and the furanose structure. In the pyranose structure, it is an hexagon with 5 carbons and 1 oxygen in the ring structure. So the fifth 5 carbons are present in the ring structure, but if we are saying hexagon, the sixth one is present outside the ring structure. Furanose. Furanose is the pentagon with 4 carbons and 1 oxygen. So here in the pentagon 5 should be there, but we are saying only 4 carbons they are present in the ring, but the fifth one is present outside the ring structure. So likewise, when they are existing in the ring structure, the pyranose form, either they exist in the pyranose form or in the furanose form. The glucose will exist in the pyranose form, and the fructose will exist in the furanose form. Now let us see the structure of glucose molecule and the ribose sugar. So those are the examples of the 6 carbon molecule hexagon and the 5 carbon pentose sugars, hexose sugar and the pentose sugars. So let us take the ribose sugar molecule. So ribose sugar, as we know that this is a ring structure exhibited by the ribose sugar. So here this is a ring structure, whereas when it is forming open chain structure, 5 carbons will be present, and the first carbon with a C double bond O and H. So OH, H, OH, H, OH, H with 2 hydrogens and OH group. So this type of organization can be seen in the open chain structure, whereas this is a ring structure formed by the ribose sugars, that is C5, H10, O5 molecule. So this is an example of open chain and ring structure exhibited by an pentose sugar, that is ribose sugar. If we take the example of a glucose molecule and hexose sugar, this also exists both in the open chain structure and also in the ring structure. So if we take the ring structure, hexagonal in its nature, with 5 carbons in the ring structure, and the fifth one is present outside, CH2OH. So this contains OH, H, H, OH, OH, H group, OH group and the H group. So likewise, this type of organization can be seen here. Here also we can see OH group. So here this is organization that we can see in the glucose molecule. This is a ring structure, whereas in the open chain structure 6 carbons are present with CHO functional group, OH, hydrogen, H, OH, OH, H, OH, H. On the last one, the hydrogens, 2 hydrogens and the OH group is present. So this is a open structure that can be observed in the glucose molecule. Likewise, both the pentoses and hexoses, both of them are showing open chain structure and also the ring structure. So here we can see that this is an pyranose. Pyranose, why? Because here it is having a hexagonal structure with 5 carbons in the ring and 1 oxygen. So this is the pyranose ring structure exhibited by the glucose molecule. Next one is a fructose molecule. So fructose is a part of ketoses, or which contains a functional group called as ketoses, or we can call it as a ketone group. So this is the one which is considered to be the fruit sugar, present in majority of the fruits except grapes, except in the grapes. So this can be seen in honey also. Honey and also in the nectar of the fruits also contains the fructose sugar. So whereas in the grapes and other one, they contains other type of sugars. So like the glucose, if you take Glucose is the one which is present in the blood, so we call it as a blood sugar. It's present in the blood sugar. So here this is showing a dextrose structure, whereas fructose is showing the Lebanose structure. The rotatory rotation is in the Lebanose rotation, whereas it is showing the dextrose type of rotation. This type of variation can be seen, and other which is called as a grape sugar also. It is also called as grape sugar. This type of variation can be seen in the fructose and the glucose molecules. And as we know that the glucose levels, the blood sugar level, if it is increasing, so that can be regulated by using some hormones like insulin like hormone, which will control the blood sugar levels in our body. In the small carbohydrate molecules, next one is derivative monosaccharides. Or derived monosaccharides we call. So derived are the one which are showing little modification from them. So let's take some of the examples. Deoxy sugar, amino sugar, sugar alcohol. So these are some of the examples of the derivatives that and sugar acid. So deoxy sugar is the one which is containing the deoxyribose sugar, and amino sugar is the one which is having the amino group in it, and sugar alcohol is the one which contains alcohol part. So this is one called as sugar alcohol, and sugar acid is the one which contains the best example is the ascorbic acid and glucuronic acid. Likewise, many such examples are there where they are considered to be having the other additional groups apart from the monosaccharide ketoses or the aldose functional groups in them. So in the small carbohydrates, next one is the oligosaccharides. Oligosaccharides are the group of carbohydrates which are having a small chain of repeated units of sugar molecules. So their number is ranging from 2 to 9 monomer units are present. Actually, it is equal to a compound polysaccharide or compound carbohydrate, equal to compound carbohydrate, but the difference is here the compound carbohydrates as like that of a polysaccharide molecule, so they should be having more than 10 or at least 10 or more than 10 monomer units. But here as they are having less than 10, so we consider them as a of course they are the one which are resembling to be like a compound carbohydrate, but these are not the part of that one, so we call them as oligosaccharide molecules. And based on the number of monomer units, so these are categorized into various types like disaccharides, trisaccharides, tetrasaccharides, pentasaccharides, etc. or different types are categorized based on the number of monomer units present in them. Let's start with the first one, the disaccharide molecules. There are many such disaccharides are there like sucrose. There are many such disaccharides are there like sucrose. Sucrose is the one which is considered to be the non-reducing sugar. Why it is called as non-reducing sugar means here it is a combination of the glucose molecule formed by the combination condensation of glucose and a fructose molecule. So the functional groups, that is aldehyde group of glucose and the ketone group of fructose, both of them have been binded with each other. So that is the first carbon of a glucose molecule and the second carbon of the ketone molecule, both of them have been binded to form sucrose molecule. So as the functional groups are not available to form a new bond with the other functional groups of other organic molecules, so we consider this as a non-reducing sugar. So sucrose is the one which is present in the sugarcane, that is Saccharum officinarum, and also in the beet sugar. A sugar beet will contain this type of sucrose molecules. So where the bonding is present between the first carbon and the first carbon of one monomer and the second carbon of the other monomer molecules. Next type of disaccharide molecule is maltose. So this is a disaccharide which is formed by the combination of two glucose molecules, glucose plus glucose give rise to maltose. So the name maltose have been derived because they are present in the malted grains of barley, and also they are present in the tissues of germinating seeds. And maltose is the one which is showing the alpha 1,4 linkages. Alpha 1,4 linkages are present in the maltose molecule. Then others are lactose. Lactose is the one which is the combination of glucose and galactose. Glucose and galactose will give rise to lactose molecule. So lactose is the one which is secreted in the mammary glands of which will secrete the milk. Mammary glands to secrete the milk. So this type of disaccharides are present, which are the combination of different types of molecules. And here also we can see the 1,4 linkages, alpha 1,4 linkages, that is between the first carbon of glucose and the fourth carbon of the galactose. The linkages are formed to form into an disaccharide called as lactose molecule. In the oligosaccharides, next one are the trisaccharides. Trisaccharides are the one which contains 3 monomer molecules, or 3 sugar molecules are present. So here in the trisaccharides, the best example is raffinose. Raffinose is the one which contains glucose plus fructose plus galactose. Glucose, fructose and galactose are present. So this type of combination is forming, that is 3 sugar molecules, or 3 monomer molecules are there. This type of combination of the 3 monomer molecules to form into a trisaccharide molecule called as raffinose molecule. Then in the same way, tetrasaccharides are there. Tetrasaccharides example is stachyose. Stachyose is the one which is formed by the combination of alpha glucose plus alpha glucose plus alpha galactose and beta fructose. So here in this one, tetrasaccharide means 4 monomer units, 4 monosaccharides should be there. So the 4 monosaccharides are 2 glucose molecules, 1 galactose molecule and 1 fructose molecule. In the same way, pentasaccharides. The pentasaccharides example is verbascose, which contains 5 monosaccharide molecules. Those are alpha glucose plus alpha glucose plus alpha glucose along with alpha galactose and beta fructose molecule. So likewise, pentasaccharide contains 5 sugar molecules. So or 5 monosaccharide molecules. The 3 monosaccharides are 3 glucose molecules, the 4th one galactose and the 5th one is a fructose molecule. So likewise here the 5 monosaccharide molecules are forming the bonding with each other to form into an pentasaccharide molecule. Likewise, hexasaccharide, heptasaccharide, all these are repeated units will be going on increasing. Such type of units, till the number of monosaccharides will be up to 9. So those are called as the oligosaccharide molecules. Next one, sweetening index. So it shows the sweetness of that particular sugar molecule which is present. So here the sucrose has the sweetening index of 100 and maltose have sweetening index of 32. Then lactose have 16. Likewise, fructose have 170. Saccharin have near about 4000 of sweetening index. Likewise, it shows the amount of sweetness of that particular sugar which is present and based on the number of monomer and also the nature of that particular monosaccharide groups which are present in the oligosaccharide or disaccharide molecules. Then reducing sugars. What are these reducing sugars? So here reduced sugars are the one in which the functional group, that is aldose group or the ketose group, can form new bonds with the other functional groups of the other organic molecules. So here if we take the example of sucrose, as we have discussed that sucrose is a non-reducing sugar. Because why it is a non-reducing sugar means, because here the functional groups, that is aldose of glucose and the ketose of fructose, both of them have been binded with each other. So there is no possibility of formation of a new bonds with the functional groups of other organic molecules. Whereas all others like maltose and lactose are the reducing sugars. Here in this aspect, so here Fehlings are the Benedict solution. Benedict solution or Fehlings reagents are the one which are useful for identifying the sugar levels in the blood. So here the one which will convert the cupric to cuprous, that is Cu plus 2 to Cu plus 3. The one which can convert the cupric form of copper to the cuprous form of copper. So that is acting as a reducing sugar. If you take a sugar molecule of any of these names that have been given here, and if you add the Benedict solution to that one, what happens is the cupric form of the Benedict solution is converted into cuprous form if it is a reducing sugar, and if it is a non-reducing sugar, the cupric form is not being converted into cuprous form. When it is converted into cupric form, it is the solution will be converted into brick red color. That shows that it is a reducing sugar. What is the significance or the function, or what is the importance of the carbohydrates or the smaller carbohydrates, monosaccharides, disaccharides, or any type of oligosaccharide molecule? So we had known about various types of the sugar molecules which are required in various levels of metabolic reactions or the metabolic functions which are taking place in the plants and also in the animals. Like if you take the example of glucose. So carbohydrates are the main respiratory substrates of the living organisms. So glucose is the one which is broken down during the process of aerobic respiration and also during the anaerobic respiration to release, is broken to release energy. So here this whatever the energy which is released here is useful for various metabolic activities in a living organism. Like for if you take the plants, for germination of the seeds, for osmotic mechanisms, for absorption of water, for various other metabolic functions, growth, development, for each and every step of the plants, this energy is required, that is coming from the breakdown of the glucose molecule. So it is providing the source of energy, and some of the carbohydrates are useful for the formation of FAD, flavin adenine dinucleotide, NAD, nicotine adenine nicotinamide adenine dinucleotide, NADP, nicotinamide adenine dinucleotide phosphate. So likewise some of the carbohydrates are useful for formation of these coenzymes which are useful for various metabolic activities both in the photosynthesis and also in the respiratory mechanisms. And in the same way glyceraldehyde, dihydroxyacetone, So these are the one which are formed in the glycolytic steps, where intermediate compounds, glycolytic step intermediate compounds. So those, if those compounds are there, they can be used in the further conversion of glucose molecule into pyruvic acid, which further be converted into carbon dioxide and water to release the energy molecules. Xylulose, erythrose, fructose, pseudoheptulose. So all these are the intermediate compounds. All these are the intermediate compounds formed in the C3 cycle, which is taking place during the carbon dioxide fixation in the C3 plants. So likewise, fructose is not only useful here, it is also one of the respiratory substrate in place of glucose molecule also. Likewise, if we take the examples of all of them, so lactose we are saying it is one which is secreting the milk, and maltose which is one which is present in the barley. Likewise, each and every the oligosaccharide or the monosaccharide molecule have significance or importance in the life of plants and also in the life of animals. In the carbohydrates, the next group are the polysaccharides. Polysaccharides are the larger molecules. It's a chain of branched or unbranched structure. So these are the one which are having at least 10 or more than 10 units of monosaccharide molecules. So these units are forming the bonds, that is the glycosidic bonds. The glycosidic bonds are formed between the repeated units of these monosaccharide molecules, and they are converted into a long chain, which may be either straight chain or sometimes they may have the branched structures also. And in these polysaccharides, so it will have the reducing end and the non-reducing end. Reducing end is present towards the right hand side and non-reducing end is present towards the left hand side. And in these polysaccharide molecules, based on the type of monosaccharides, whether the polysaccharide is made up of only one type of monosaccharide or different types of monosaccharides, so those are of two types, homopolymer molecules or the heteropolymer molecules. Let's start with the homopolymer molecules, or the polysaccharide homopolymer polysaccharide molecules. So homopolymer means the one with the polysaccharide molecule or an complex molecule, complex organic molecule, which is formed by the repeated similar type of unit molecules, or similar type of repeated units are present. So by forming a bonds between them. So this type of structures are called as a homopolymer molecules, like cellulose, glycogen. Such examples where a homopolymer which are made up of repeated units of glucose molecule. Repeated units of glucose are present in them. First one, homopolysaccharides. Homopolysaccharides. So homopolysaccharides are the one which are made up of similar type of repeated units. So in this also, they may be either made up of similar units, like repeatedly glucose molecule may be present in the entire homopolysaccharide. So those are called as glucons. So the one which contains only fructose, fructons. The one if they have only xylose, xylons. So likewise galactons. Based on the type of the repeated unit in which they are by which they are made up of, they are glucons, fructons, xylons and the galacton molecules. And heteropolysaccharides. Heteropolysaccharides are the molecules here different repeated units are present. different repeated units are present, where it may be made up of glucose, fructose, or fructose like repeated units of the galactose molecules, xylose molecules, or different types of repeated units are present. That is called as a heteropolysaccharide molecules. So these repeated units are binded with each other by the glycosidic bond with a linkage called as alpha 1,4 linkages. If the side chains are present, the side chains are linked with the alpha 1,6 linkages also. So likewise, based on these linkages or by formation of these linkages, they are forming into a long chain of polysaccharide molecules. In the polysaccharides, the first one is starch. Starch is one of the most important food content present in the human beings. So this is taken by the human beings in different forms like cereals, right, in the form of rice, wheat, and some pulses also have the starch, and potato, banana are some other examples where the starch content is very high, which is consumed by the human beings, which will be useful for producing energy by the digestion process. So likewise, this is one of the major constituent part present in the human digestive system. And if you take the content related to the plants are, as we know, they will perform photosynthesis. By photosynthesis, they will prepare their own food materials by using the raw materials. So that is by using the raw materials like carbon dioxide and water in presence of sunlight and chlorophyll molecule. So they will convert these raw materials into the photosynthase, that is carbohydrates or the polysaccharide molecules. That is the starch is synthesized by this process. So here, whatever the starch which is synthesized by the plants, so in which form does this starch is stored in the plant cells? So is it stored in the cells or in any part it is stored? Actually, the plants which are producing the food materials, so they are storing the storage of starch. So the starch is stored in various forms. So either it may be in the cytoplasm or in the cell or in other cell organelles, like in the chloroplast where it is synthesized, that is starch grains we call it as. So otherwise, if it is stored in the cells, so it is stored in the cells in the leucoplast. So due to presence of the leucoplast plastids, so that part of the cell will store the food materials. So the one which are storing the starch storing chloroplasts are called as amyloplasts. So this type of nature can be seen in the cells, and here we are saying the starch grains are formed. So here the starch grains are for acquiring different shapes when they are forming into grain like organization. Either they may be round, oval, or cylindrical or rod shaped also. Likewise, different shapes are formed. So like oval, round, rod shape, etc. Shapes will be formed. When the starch is forming into different shapes, so they are formed around a central point. That central point is called as hilum. Around the central point, the concentric layers of the starch will be formed. So likewise, we can see around this one, the concentric layers of starch will be formed. So this type of structures are produced because of the layering form of the starch which is formed here. So here the simple structures may be formed or compound structures may also be formed. So this is the compound starch grain which is present in potato, whereas compound structures are also being formed in potato where this type of organization can be seen. Where we can see concentric arrangement of or eccentric arrangement can also be seen here sometimes. So this type of organization is called as a compound type of starch grains in potato plant. The layer formation may be either in the concentric form. So concentric means the starch layers are formed around a central point called as hilum in a concentric or the round form of ring-like structures will be formed. whereas in the eccentric form they are not formed in a one single structure. So the type of organization is called as a eccentric form. So that the rings, either the concentric or the eccentric form of starch layers will be produced. Whereas in the pisum, if we take, so in pisum, so the central point will be like this. So around that the layers of the layers of starch will be produced like this. So this type of can be seen in the pisum plant. So likewise in different types of plants, this is a hilum point, the central point will be there, this is a hilum, and these are the layers of starch. So likewise in different shapes may be acquired in different types of plants in the starch grains. Starch is a structure or carbohydrate or the polysaccharide molecule which is formed by two types of units. So those are called as amylose and amylopectin. These two repeated units are present in the starch structure. So amylase accounts for about nearly 20 to 30% of the starch composition, whereas the remaining amount or remaining content is occupied by the amylopectins. So that is large proportion is made up of amylopectins. So here if we take the cereals and other pulses or other grain like structures, so in them the majority entire content is made up of amylopectin. Whereas in the pea like examples, cereals like in maize, this type of nature can be seen. Maize. In pea, the wrinkled surfaces are present. The wrinkled surface is made up of near about 98% of amylose. So this type of variation can be seen in the content of amylose and the amylopectin present in the starch of different types of plant species. Amylose is a structure which contains nearly 100 to 200 glucose molecules in it. Repeated units of glucose are present. So here it is an straight structure which is formed by 100 to 200 glucose molecules, but here the straight structures may produce helices here and there. So here in the helices structures, near about 6 the helices are formed at every 6 glucose molecular structure. So if you see the structure, so likewise we can see each one is an one glucose monomer molecule. So likewise the folding can be seen at every 6 points of the glucose molecules. Likewise helices will be produced. So this type of organization can be seen in the amylose where 1,4 linkages are present. So here between the glucose molecule we can see alpha 1,4 linkages. So what are these alpha 1,4 linkages means? So the glucose molecules are linked with each other by glycosidic bonds. So these glycosidic bonds are formed by alpha 1,4 linkages. That is the carbon 1 or the first carbon of the glucose molecule is binded to or is binded with the 4th carbon of next glucose molecule. This type of linkage is called as a alpha 1,4 linkage. So because of this alpha 1,4 linkage, it will be forming a straight structure with a helix like organizations here and there, and each helix contains 6 glucose molecules. Next one, amylopectin. Amylopectin is the one which is made up of nearly 2000 to 20,000 glucose molecules. 2000 to 20,000 glucose molecules. Here it is also a straight molecule. Where here the glucose molecules are showing 1 alpha 1,4 linkages. Alpha 1,4 linkages are there. So here if you see the structure, each one is a one glucose molecule. each one is one glucose molecule like here. So here the round structure is a glucose molecule. Each glucose molecule is binded to the adjacent glucose molecule by 1,4 linkages. That is the same type of linkage that we had seen in the amylose, where the first carbon of one glucose molecule is binded to the fourth carbon of the adjacent glucose molecule. So not only here the straight structure, it will have the branches also. Side chains are also being seen here. The side chains are also being formed by the same glucose molecules. These are the side chains which have been formed by the glucose molecules, and here wherever the side chains are present, that side chain is forming a linkage called as 1,6 alpha 1,6 linkages. So what is this 1,6 linkage means? The bonding, the glycosidic bond is formed between the sixth carbon of glucose in straight chain. So straight chain means this one, the glucose of this straight chain. The sixth carbon of glucose in the straight chain is binded to first carbon of glucose of side chain. That is, let's take the example here. This is a glucose, this is a one glucose of straight chain, this is a glucose of the side chain. The first sixth carbon of glucose of this straight chain is binded to the forming a glycosidic bond with the first carbon of the glucose of the side chain. So here also the same thing. So here also the same thing we can see here. This type of linkage, that is here we can see both the alpha 1,4 linkages within the straight chain and alpha 1,6 linkages with side chain present in the branched structure. So likewise amylopectin is showing little bit complex structure when compared to the amylose molecule. Amylose give blue black color with iodine solution. So actually the starch are tested with iodine solution. When the amylose is present, the iodine will be converted, whenever iodine is added to the amylose part, so that will be converted into blue black color. Whereas if amylopectin is there, so amylopectin is identified based on the red violet with iodine solution. So likewise this is a test to identify the presence of amylose and the amylopectin in the food content, or we can say that starch is tested by iodine solution. So when the iodine solution is added to the starch molecules or to the starch food material, if amylase is present, that food material will be converted into blue black color, and if it is amylopectin, it is converted into red violet color. In the polysaccharides, the next one is glycogen. Glycogen is the abundant the type of polysaccharide which is present in the animal bodies, particularly in the microbes, particularly in the bacteria we can say. They are present in the bacteria and some of the animals, and here the glycogen is made up of nearly 30,000 glucose molecules. Repeated units of glucose are present, near about 30,000 are present here, and here these 30,000 glucose molecules, because of this it is showing near about 4.8 Daltons of molecular weight. Glycogen is a structure which is showing the straight chain and also the side chains. So we can say that it is showing the branched structure. In the straight chain, we can see alpha 1,4 linkages. So what is this alpha 1,4? Once again the same type that we have seen in the starch. That is the first carbon of one glucose is binded with a glycosidic bond with the fourth carbon of the adjacent glucose molecule. Likewise alpha 1,4 linkages are present in the straight chain, whereas in the side chain, with the side chain we can say alpha 1,6 linkages are present. So that is glycosidic bond is formed between the sixth carbon of glucose present in the straight chain with that of the first carbon of glucose present in the side chain. Likewise, we can see both 1,4 linkages and also the 1,6 linkages. So, let us see the structural organization with the help of an diagrammatic representation in the glycogen molecule. So, in the glycogen, at about every 10 to 14 sugar units, the side chains will be arising. Whereas in the amylopectin, we had seen that at every 20 glucose molecules, the side chains will be arising, but here at every 10 to 14 glucose molecules, the side chains will be arising. So, here we can see straight structure along with the helical like organization. So, like this 6 glucose molecules which are forming into helix like structure, and once again the straight structure to some extent, and later once again forms a helix. So, likewise we can see straight and also the helix structures in the basic structure of this glycogen. Apart from this one, it also have the side chains like this. The branching is also being seen. So, side chains and also the branching like structures are seen like this. These side chains or branching like structures are formed at every 10 to 14 distance of 10 to 14 glucose molecules. This type of complex organization can be seen in the glycogen molecule. So, this is a basic schematic diagram representing the glycogen molecule. So, here the bond that is present between this glucose and this glucose will be 1,4 linkage, and this glucose and this glucose will be the 1,6 alpha 1,6 linkage. We have seen the schematic diagram, but if you see the actual the ring like structures which are formed here. So, this is a glycosidic bond. Whenever the glycosidic bonds are formed, they are formed by releasing of one water molecule. So, these are the glycosidic bonds here, and these are the glucose units, glycosidic bond. In each of them, so first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and here this is a sixth carbon, right. Here also the same thing, like first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and CH2OH. So, as I am saying that it is a straight chain. In the straight chain, the 1,4 alpha 1,4 linkages are present. That is, the first carbon of one glucose molecule is binded by glycosidic bond with the fourth carbon of its adjacent glucose molecule. In the same way, here also the first carbon of this one with the fourth carbon of its adjacent one. So, once again the glycosidic bond. First, second, third, fourth, fifth. Here, when a side chain is formed, so now here the sixth carbon will be present. The side chain is formed between the sixth carbon of the straight chain and the first carbon of the side chain. Like this is a side chain. The first carbon of the side chain. So, like this the bond will be formed. So, second carbon, third carbon, fourth carbon, fifth carbon, and of course the sixth carbon here. And here once again we can see glycosidic bonds. The first carbon, second, third, fourth, fifth, and once again sixth carbon. So, here in this chain also we can see 1,4 linkages, but wherever the side chain is arising, at that point only here the 6 and 1 and 6, that is alpha 1,6 linkages are present. Whereas further in the side chain alpha 1,4 linkages are present. And in the straight chain also everywhere we can see alpha 1,4 linkages. So, this type of diagrammatic, of course, if you take this part also, it is a continuous straight chain like this. So once again 0, O. So likewise 1, 2, 3, 4, 5 and the sixth one. So it is a straight chain and it is a side chain. So wherever the, for example, if the side chain is formed here, so instead of this CH2OH, what happens? So one more side chain will be formed with the sixth carbon with that of the first carbon of the side chain. Likewise, this type of structural organization can be seen in the straight chain structures of a glycogen molecule. Next one is inulin. Inulin is also a type of polysaccharide molecule. So this is present in the root tubers of dahlia. Root tubers of dahlia, and here these are not metabolized in the human body, and this inulin is directly filtered through the kidneys. So these are the one which are useful for testing the kidneys. So how much it is filtering the things. So that is for the testing of the kidneys, the inulin will be used. So this is also one of the important, not major, so repeat. So inulin is also one of the important type of polysaccharide, which is the food storage structure, or the one which is used as a food in various animals and also in the human beings. Next one is chitin. Chitin is a structural polysaccharide molecule which is present in the exoskeleton of arthropods, particularly in the cockroach like examples, and also in the cell wall of fungi. Normally when we are saying that polysaccharides, so they are having the repeated units of glucose molecule, but here the glucose is not present, but it contains nitrogen containing glucose, and here they are forming a N-acetyl glucosamine. Repeated units of N-acetyl glucosamine is present. Repeated units of N-acetyl glucosamine, and these are showing alpha 1,4 linkages, the same type of linkage where the first carbon of earlier monosaccharide molecule is binded to the fourth carbon of the adjacent monosaccharide molecule. So this type of structure can be observed in the chitin also. This type of structure is observed in the chitin, which is present in the exoskeleton of cockroach and the cell wall of almost all the fungi. Generally fungi, we consider fungi as a plant, but so it is not a cellulosic one, which should be made up of the glucose molecules, but instead of that one it contains the nitrogen containing glucose repeated units, that is called as a N-acetyl glucosamine, which is present in the cell wall of fungi, which is a deviation from the normal cell wall present in other plant cells. So chitin is generally soft and leathery. Because of this one, it is providing strength and elasticity to the cell wall which is present in the plant organization or in the fungal organization, and here and also in the animals wherever it is present. If it contains proteins and other calcium substances, it will becomes hard. So the type of nature will be there related to the chitin polysaccharide molecule. So here in this one, as I said, that it is a unbranched structure. Here the residues, the glucose residues are present. These residues are at angle of 180 degrees. So this type of nature will be there with 1,4 linkages, and this type of nature, let's see in the form of a diagrammatic representation of the residues of glucose or derivatives, that is N-acetyl glucosamine molecules. So these are the N-acetyl glucosamine molecules where NH COCH3 are present, and CH2OH. So this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth carbon. Like these are also like the pyranose molecules as like that of normal glucose structures. So in this one, the glycosidic bonds are formed by dehydration process. So in this form, in this structure, so here we can see NH, CO, CH3, and CH2OH here. Then the 180 degrees of variation can be seen here with once again NH, CO, CH3, and CH2OH. So here the one which is present here, it is quite repeated, it is showing a 180 degrees of curvature with 1, 2, 3, 4, 5, and the sixth one. So here the first one, the second one is here it is showing the 180 degrees of exactly the curvature. So that that's why we say 180 degrees of curvature is present. But here once again 1, 2, 3, 4, 5, and sixth one. So likewise from here to here also once again it is showing the 180 degrees of curvature. So this type of organization which is unbranched with 1,4 linkages, that is the linkage is present between the first carbon of first N-acetyl glucosamine with the fourth carbon of next N-acetyl glucosamine. Once again the same, first carbon of this glucosamine and fourth carbon of this glucosamine. This type of organization can be seen in the chitin molecule. So next one is cellulose. Cellulose is one of the polysaccharide molecule which is present most, which is considered to be the most abundant one present in the various parts of the plant cell. Majorly it is present in the cell wall, because major constituent of the cell wall is made up of this cellulose only, and which accounts for 50% of carbon present in a plant cell. So and 90% of the other 90% of the major constituent part is made up of the cellulosic carbon content. So here this is this contains 0.5 to 2.5 Daltons of molecular weight, which are having 6,000 units of residues of glucose molecule. So this is also a this is also made up of repeated units of glucose molecule. Here it is also an unbranched structure. Cellulose will account for about 20 to 25% of the compositional dry weight of the plant body, because the cell wall is a major constituent structural unit. So cellulose is also one of the major, or we can say this is the major constituent part of the structural unit of a plant cell, because cell wall is a major structure by which this cellulose is by which the cell wall is made up of this cellulose. In cellulose also the angle is of 180 degrees, where if you see the structural organization where the repeated units of glucose are present. So this is the furanose structure of the glucose. The first carbon, second carbon, third carbon, fourth, fifth carbon, and the CH2OH group is present. So where this is forming a glycosidic bond, as we are every time saying that glycosidic bond is formed by the dehydration process, that is loss of water molecule during the formation of glycosidic bond in the structures. So here the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth carbon. So here alpha linkage, alpha 1,4 linkage is present, that is the first carbon of the one glucose molecule is forming the glycosidic bond with the fourth carbon of its adjacent glucose molecule, and here it is the O group is here, as whereas O group is here, that is showing, it is showing a 180 degrees of this curvature. Then once again one more glycosidic bond is formed here, So first carbon, second, third, fourth, fifth, once again the sixth carbon here. Once again here the glycosidic bond, that is alpha 1,4 linkage is formed. So this type of straight chain without any branches can be observed in the cellulose molecule. So this type of structures can be observed in the cellulose, and cellulose is a major constant part of the cell wall and a structural part of the plant body, which is very much required for formation of various structures, and it is also being present in the wood, the bark part and also the cork tissue part. Cellulose is consumed by the animals and also by human beings, but in human beings it is not being digested because amylase which is present in the digestive system is not able to digest the cellulose content of the food which is consumed by the human beings. But in the ruminants contain some bacteria inside their digestive tracts. So this bacteria is having the ability to digest the cellulose. It is having the ability to digest the cellulose. This type of nature is present in the ruminant animals. Next one, cellulose nitrate is formed with the help of this nitrate, with the help of the cellulose. The nitrate is combined with the cellulose to form cellulose nitrate, which is used as an explosive. And some of the cotton fibers are linen fibers, so which are useful for clothing purpose, which are also made up of the cellulosic major content is made up of the cellulose. Then some other fibers are there which are the mixture of various types of cellulosic content with the others one, which are useful for various purposes. Like if you take the jute fibers, and apart from that one, the other fibers are there which are useful for various other commercial purposes for the survival of the humankind. Mucosubstances. Mucosubstances are the mucilaginous or slippery like nature. So these are made up of uronic acid, galactons, mannans, etc. So like these are the one which are having the slippery nature, or we can say some mucilaginous structure. So this mucilaginous nature is helping protecting the plant surface from various other side effects or various other harmful effects from the environmental structures. So likewise these are being synthesized inside the plant bodies and also on the surface of the plant bodies, like mucilage on the outer surface of some algae groups, like in the nostoc and anabina like one, they have a mucilaginous substance outside them. So that is nothing but the protective structure which from the other environmental harmful effects. Then agar-agar. Agar-agar is a chemical which is extracted from red algae like Gelidium and Gracilaria. So this agar-agar is used in the tissue culture. So actually in the tissue culture process, we know that culture medium is prepared, which is in a liquid form. So in order to solidify that culture medium, so this agar-agar is added, so that on the solidified culture medium we can introduce we can inoculate the explant, so that by absorbing the nutrients from the explant. So by absorbing the nutrients from the culture medium, the explant will grow into a small plantlet. Then carrageen. So this is the one which is also been extracted from some algal group. This is also one of the mucosubstance. So likewise many such mucosubstances are there which are useful for the plants, protecting them from the other harmful effects. And apart from that one, some of them are useful for humankind also, like agar-agar. We can say that it is used in the making of jams and jellies also. Pectins. So pectins are the substances which are the part of the cell wall, major which is considered to be the made up of galactose, mannans and galacturonic acid. So like that these are the substances which are present in the cell wall and concerned with the binding like structure in the cell wall organization. And peptidoglycans. Peptidoglycans are the structures which are present in the bacterial cell wall. So these are made up of repeated units of N-acetyl glucosamine and N-acetyl muramic acid. So these are generally called as NAG and NAM. So which is protecting the bacterial cell from various harmful effects, and also from protecting from osmotic lysis also. Likewise, various major significances are related to the peptidoglycans which is present in the cell wall of bacteria. It is otherwise called as mucopeptides or the murines or muramic acid also. Next one, hemicellulose. Hemicellulose is a structure which is made up of xylons, galactons, arabinose and mannose. So which is acting as a cementing like or binding like structure between the cellulose and the pectin substance which is present in the cell wall of primary cell wall structure, which is acting as a binding structure for the pectin and the cellulosic microfibrils present in the primary cell wall structure. These are some of the examples related to the mucosubstances present in different types of living organisms.